So we're celebrating today science. We don't have a lot to celebrate, so this is a bit of light in a very dark period. But this is the World Cancer Day, and even though we have some really grim numbers, looking at 10 million people that died of cancer each day in the recent years, and 20 million new cases of cancer in a year, we know today that 30 to 50% of all cancers are preventable. 40% of cancer-related deaths are linked to modifiable risk factors. Prostate and breast cancers, male and female respectively, continue to lead the incidence of new cases of cancer, and the lung continues to be the first reason for death from cancer. We see some changes in the incidence and epidemiology due to early diagnosis and also due to new therapies and immunotherapies that are really changing the landscape of cancer. Of all cancer deaths in people under 50, for example, colorectal is now number 1 in men and number 2 in women. So for the young ones in the audience, this is a field probably to invest in the future. The estimated number of new cases in 2040 is 30 million. This is a really good number, thinking that we are investing so much in cancer. But together with that, if we look at how many numbers of approvals the FDA went through in the last year, we see that the new anti-cancer drugs constitute 24% of new approvals, and the 5-year average for cancer approvals is 28%. So looking at these optimistic numbers, this is what we're celebrating today. Thanks to the wonderful lineup of speakers today, of scientists and clinicians coming from Tel Aviv University, the Weizmann Institute, Ben-Gurion University, the Hebrew University, Bar-Ilan University, and the Technion in Haifa. Also coming clinicians coming from hospitals, from Sheba Medical Center and Schneider Children's Medical Center. So I thank you all, and I would like to start with a welcoming note from our president, Professor Ariel Porat. Thank you for coming.
Thank you, Ronit. Dear friends, colleagues, while we are speaking here, a war is still going on between Israel and Hamas following the terrorist attack which took place 4 months ago when 1,300 Israelis were murdered. Most of them are civilians, including babies, young children, women, men, old people, sick people, and 240 people were abducted. Until now, there are still more than 130 hostages who are held in Gaza by the Hamas, and we all pray for their return home quickly and safely. Until they return, the wound that inflicted upon us on the 7th of October would remain open. So we hope for release of the hostages, as I said, as soon as possible. During wartime, maybe paradoxically, we think about human welfare. We think about human welfare of our soldiers, of the people who were injured. We think about, of course, the hostages, about the welfare of the people who were evacuated from their homes, and even about the welfare of people on the other side of the border who are not involved with the Hamas. And welfare is also, in a way, the topic of today's conference, because human welfare. And as Ronit mentioned, gave you a few some data about the cancer disease or cancer diseases, actually. There are 300 of them, something like that, right? We know that about half of the people suffer or will suffer from cancer during their lifetime. You know that about 1/3 of the people would die from cancer, so there's no better goal for combating this terrible disease. And this annual conference for cancer research and clinical research, basic research and clinical research, is held by the Cancer Biology Center at Tel Aviv University, led by Ronit, Professor Ronit Satchi-Fainaro. The center has some other initiatives and centers acting under this center. To mention one is the Varda and Boaz Dotan Hemato-Oncology Research Center, and there are others. Tel Aviv University has several characteristics that are also reflected at this center, the Cancer Biology Center. And the first one is the international aspect of the university and also the international aspect of the center. Even though the conference is led by professors and researchers from Israel, from all universities, the members, the people, the researchers from Israel and from Tel Aviv University acting under the center, collaborating with their colleagues all over the world. And needless to say how important it is, especially those days when Israel is a little bit isolated comparing to regular times. The second characteristic of the center, another characteristic that Ronit mentioned, is the multidisciplinarity of the work done under the center. So the more than 100 research groups acting under the center are composed not only of scientists coming from the Faculty of Medicine or the Faculty of Life Science, but also from engineering, bioengineering, and electrical engineering, but also for computer science, chemistry, physics, and other related fields. And that's what makes the center a unique one, and the operation within Tel Aviv University is very natural, because also Tel Aviv University is such a comprehensive university that includes all the relevant fields for research. The third, and maybe the most important characteristic of the center, is the collaboration with the hospitals, 17 hospitals affiliated to Tel Aviv University. It's about 50% of the market share in terms of number of patients in Israel. And the collaboration with clinicians, with doctors, and with researchers at the hospitals makes the collaboration very fruitful and important, because even though we appreciate very much basic research, we also want to bring translational medicine into the world, and the center's goal is both basic research but also applications and making the world better, enhancing human welfare, as I have mentioned before. Maybe last word about the situation we are in. So, Tel Aviv University opened the academic year just a month ago, after postponement of about 2.5 months, because we waited for the soldiers, for the reservists to return to campus. And even now there are still thousands of students who are in actual duty. And the reason that the university decided to begin the academic year, even though some of the soldiers are still not here with us, is for two reasons. One is that we understood that if we don't do so, we would jeopardize the academic year and everybody would suffer, including the reservists. But no less important, it's because of the resilience of Israel. Israel, even though while conducting a war, could not stop the economy, and also we could not stop producing doctors, we cannot stop producing scientists, we cannot stop producing engineering and intellectuals, etc. And for that reason, the university decided to return to normalcy, even though we are still not in normal times. I think that this conference also represents the spirit of Tel Aviv University, that research must go on, especially in such an important field as combating cancer. Thank you very much, and enjoy the conference. I'm sure it will be a very fruitful one and enjoyable too. Thank you.
Thank you, Professor Porat. Without further ado, we'll start with the first speaker, Professor Yardena Samuels from the Weizmann Institute of Science, who will speak about revisiting the neoantigen approach for cancer immunotherapy. Welcome.
Okay, so you already noticed I changed my title. So I'll be talking about first of all, thank you very much for the invitation. It's good to be here. So I'll be talking about the role of tumor heterogeneity in immune evasion. So we all know that mutations drive cancer progression. We see here a clear phylogenetic tree of various somatic mutations developing at a stage-wise manner in cancer. But we need to remember that cancer progression is also affected by immune rejection, mostly by the interaction of the presentation of an antigen on an MHC molecule and a T cell receptor on the T cell, and we'll get back to this later in my talk. We know that the main biomarker for immune checkpoint inhibitors is tumor mutational burden. This has been seen again and again, seeing as TMB will derive highly mutated proteins, some of which will be presented on the MHC molecules of the cell. Another strong biomarker for immune checkpoint inhibitors is low heterogeneity, meaning highly clonal neoantigens that are being presented on tumors. This was presented by the Swanton lab a few years ago. And so my lab decided to try and tease apart these two attributes of TMB and clonality. And so Osnat and Yochai in my lab derived this model system where they took melanoma cells, exposed them to UVB, which made the cells highly heterogeneous and highly mutated, high mutation load. And to our surprise, this high mutation load, which is supposed to derive a lot of presented mutated neoantigens, was actually extremely aggressive when you grew them in an immunocompetent mouse model. But when they took single cells from this population, we now had a low heterogeneous cell line, and the mutational load was random. And when they injected these cells 20 times with 20 different cell clones, each time they were rejected. And we see this right here. So ultimately, what we found out was that if you have a high intratumor heterogeneity tumor, it will actually be extremely aggressive due to low immune infiltration, low antigen-specific T cells that are found there, whereas if it's a low ITH, the opposite will occur. And the take-home message from this study was that it's worth identifying tumor intratumor heterogeneity, that it is an additional biomarker for aggressiveness of a tumor, as well as response to immune checkpoint inhibitors. And this quantification is highly relevant. So we decided to follow up on this study, really focusing again on low and high ITH in tumors. And what we can see here is, despite the fact that we still think that high ITH tumors are highly aggressive, and the low ITH tumors are less aggressive, when you closely look at the low ITH tumors, you can see that 40% of them actually exhibit a poor outcome. They have a lower survival time than the high ITH patients. And so we think that there are additional factors that contribute to immune escape of these low ITH tumors. And so we decided to focus on these and try and understand it better. So we wanted to identify mechanisms responsible for immune escape of tumors that are bearing low intratumor heterogeneity. So we derived a new phylogenetic tree, meaning we took a UVB-exposed cell line, derived single-cell clones from that cell line, performed whole exome sequencing based on their genomic attributes, distributed them across the tree, evolutionary development, and you can see that we have here several branches with the different clones that we derived, and we decided to focus on one particular branch that had 6 single-cell clones. And the reason we chose this is because we had sufficient number of single-cell clones here to start comparing them, and they also had different phenotypes, so we're pretty striking. You see here on the left that we have 3 single-cell clones that are aggressive, whereas 2 that get rejected. Now, we expected all of them to be rejected, right? Based on our previous study, all the homogeneous single-cell clones were rejected. Here we're finally finding some that do not get rejected, which depict the patients I told you about, those 40% patients at a low ITH and are still aggressive. So we thought this is a perfect model to start investigating attributes that lead to this aggressive growth. So when we further looked into these clones, we could see that they're actually highly similar genetically. You see the variant frequency here, so they're highly clonal, just as we expected. When we inject the cells that are rejected into immunocompromised cells, you see that they grow similarly. So this has to do with the immune system. And when we start comparing their whole exome sequences to see if the differences between them has to do with genetic changes, we only see one somatic mutation that differs between all these single-cell clones. And if this is a neutral mutation, it probably doesn't give the reason for the difference in phenotype. So we're thinking this must be due to epigenetics, differential expression genes. So we wanted to further investigate this, and we did this very large study where we injected these different single-cell clones, the ones that get rejected, the ones that are very aggressive, took these tumors at different time points and performed single-cell RNA-seq to start comparing their expression differences. And so with Answer Satpathy and Stanford and Joy, we immediately could see when we collapsed these different immune microenvironment cells in these different clones, you could see over time this very major change in the different cells that infiltrate the tumors. And the main one that showed a difference is tumor-associated macrophages, which infiltrate specifically the clones that are non-rejected. Okay, the highly aggressive clones. And this is just a quantifying part of the analysis. And you can see this is very, very significant, specifically for TAMs. So we further looked into the CD14 positive subpopulation and subdivided them into 6 different clusters. And here again, when you look over time, you can see 2 particular clusters that are enhanced, specifically in the non-rejected clones. And when you look at what attributes these particular clusters have, immediately you can see that they're high for the M2 score. So not only do we have these TAMs and macrophages that are infiltrating tumors that are highly aggressive, these are also harboring an M2 score, and they're also very high in expression for complement genes. C1q and C1q A, B, and C, and ApoE, which has been seen in the literature. So we're not the first to see this correlation to complement increase. The additional attribute that we saw in the non-rejected clones is that they had less T cell infiltration, and that the T cells that did infiltrate were exhausted. They had a high exhaustion score. So it all made a lot of sense. So we obviously now validate this single cell data using OPAL, and we validated them. We can see here the CD206, CD204 pro-tumorigenic macrophages that are infiltrating specifically the non-rejected single cell clones, and this is a significant result, and a lower CD8 positive T cell infiltration in the non-rejected clones. So we wanted to understand the mechanism behind this, and so we started looking at the differential expression of the various genes across the different clones, across the different time points, and we see here a lot of data. We wanted this to be consistent across the clones and the time points, and the one that really we noticed immediately is this target called MIF. And so we decided to further look into it. These are just the summary of the different, because of course there are many additional targets that seem interesting, but macrophage migration inhibitory factor was the one that seemed most interesting. So we further focused and decided to perform an in vivo CRISPR screen where we took all the targets that were consistently differentially expressed between the rejected and non-rejected clones over time and reanalyzed them in vivo by taking a cell line that was not rejected, infecting it with these different targets, injecting these tumors into an immunocompetent mouse model, checking their population at the beginning, and then taking the tumors at the end and comparing which remained at the end. And then check really which were depleted towards the end. And immediately we could see that MIF was the gene that was depleted and enriched. And so we decided, okay, this is an additional reason to focus on MIF beyond looking at human data as well. So with ATAN, we looked at MIF expression in melanoma patients and the correlations to survival. And you can see that high MIF expression has a high correlation with low survival. And this is even enhanced if you start looking at T cell infiltration. So again and again, there's this correlation of high MIF expression that significantly associates with poor patient survival and a high and low T cell infiltration. So this is our model. We have melanoma cells that produce in MIF. MIF is a secreted factor. It binds CD74. This is a receptor that is found on various different cells, but also macrophages, M2-like pro-tumorigenic macrophages, which fits what we saw with the single cell data. And so we think that then these macrophages are recruited, inhibit, and they're inducing tumor growth. Now, MIF has been seen to be an important inflammation mediator, playing a role in tumor aggressiveness, but we also see contradictory results in the literature. So we think that our model system actually gives us a better insight into what MIF does because it's so homogeneous. All of these studies were done in a very heterogeneous environment. So we went ahead and knocked out MIF in our various clones using CRISPR, re-injected these cells, and you can see how dramatic the results. The moment you knock out MIF, you lose the aggressiveness of the tumor growth. And then again, we went back to do the single cell RNA-seq on these different tumors. You see here the t-SNE plots where again you see a very nice result where we have the MIF clones, they're less aggressive as expected, and then you also have low tumor infiltration of TAMs. And this is the quantifying part of it. And you see it's highly significant. So the fact that we knocked out MIF was able to revert also the phenotype of TAM infiltration. Again, we performed the CD14 positive M2 score. It reverted the M2 score. It reverted the expression of these complement genes, and it also reverted the infiltration of T cells. So now when you have the knockout clones, you have better infiltration of T cells and the exhaustion score is gone. So, getting all of these consistent results is not obvious. We know that. So we're very happy because there was a lot of work invested in knocking out MIF. Again, we validated all this data on the protein level using OPAL. So again, these are the macrophages that were stained, and we reverted and as well as the T cells, which reverted their phenotype. So at this point, we have this model. We have the non-rejected melanoma cell, which is, again, it's highly homogeneous but highly aggressive, has a high expression of MIF. MIF, if secreted, it binds CD74 on the M2 macrophages. It increases the immunosuppressed microenvironment, which leads to further growth of the tumor. There are inhibitors to MIF, such as ISO1, and there are additional inhibitors that have been developed, and we think that it may be worth testing these inhibitors on this particular model system, to see if we can now revert this phenotype. So this study is as yet unpublished, it's in revision. So at the end, if anyone has ideas, criticisms, we'd love to hear. I'd love to hear. This is just a switch to a different direction that I have in my lab, which is looking at neoantigens. So, as I said, the interaction between the cancer cell and the immune system, in this case specifically T cells, is extremely important in immunoediting. This is derived through neoantigens, so proteins that harbor a mutation and therefore non-self rejected by the T cell. You can see here this interaction, and it's essential in order for immune checkpoint inhibitors to work. So we're interested in identifying these neoantigens. The way we do this is by immunopeptidomics, basically by a chemical assay where we elute peptides from the MHC and then analyze them and identify them on the surface. So it's very important to, beyond identifying neoantigens, we see here a lot of black marks. This is the dark matter, a lot of unidentified neoantigens. We want to know what they are. They're very important because more than 50% of responders to immune checkpoint inhibitors are low TMB, meaning that there are additional sources for antigen presentation. We think that there are many sources. These are the hallmarks of antigen in cancer. So we think one of these dark matter are bacterial mutations. This is a study we published with Ravid, Straussman, and Jennifer Wago, where we found bacterial-derived peptides, some of which are immunogenic, presented on melanoma cells. Another category of dark matter, non-canonical presented peptides are derived from the translation operations. Without going into detail, because this is already published, we have identified peptides that are derived from translation aberrations, mistakes in translation. And these are also immunogenic. And we're thinking of this as an additional level of intratumor heterogeneity. And this is where I will leave you today. This is my group, amazing, amazing group, who keeps working despite the very difficult situation. And then our many, many collaborators. Thank you very much.
Thank you for this really exciting presentation, fascinating findings. Angel, we have some time for questions, yes.
Yeah, that's a good idea. So we're doing these experiments. We just got the review comments, and maybe you're one of the, but someone asked about correlation of expression of MIF and the other clones, and so we're now trying to see that correlation. So thank you. Yeah. Yeah, so I'm pretty sure that we did look at aneuploidy. We're always happy to revisit it, but that was part of the genomic analysis to assess what is driving this. Regarding epigenetics, so actually Gil Bendek, apparently from Hadassah, he works on MIF, and in his postdoc he looked at polymorphisms in the promoter of MIF that affect its expression. And the way I found out that it was when we put this in bioarchives, and he contacted me. So now we're looking to see if anything, the promoter of MIF, and maybe there's a difference between these different clones or particular transcription factors that may affect different levels of expression of MIF. So that's under investigation. Yeah, it's a good question. So the way we try to, and I didn't put it here, what we did is what we like to do mixing experiments, right? So we take single cell clones and we mix them with the original, and we put them in 10%, 20%, 50% in order to increase heterogeneity and see what happens. Seeing as this is a secreted factor, we expect that there will be a tipping point in which the phenotype would be affected. Indeed, that is what happened. So at a certain percentage where you do the mix, suddenly you revert the phenotype of the heterogeneous and vice versa. The homogeneous is no longer non-aggressive. So we played around that. And so it means that if you were to go back to the human data and wanted to use this as a biomarker, you would need to check how much MIF is expressed and beyond which threshold is it going to start having an effect. But that we haven't done at all.
Thank you.
Thank you.
Thank you, Yardena. Our next speaker is Dr. Ronnie Shapira from Sheba Medical Center, who will speak about adoptive cell transfer with tumor-infiltrating lymphocytes, TILs, for solid tumors, current role in checkpoint blockade era. Thank you, Ronnie.
So I'm a medical oncologist, and
you'll hear some of the clinical insights of treating patients with advanced melanoma and other solid tumors, some of the struggle to improve cell therapy for patients with metastatic melanoma and other solid cancers. So if we speak on adoptive cell transfer with tumor-infiltrating lymphocytes, it's not a new concept. It's almost 30 years now that we speak about clinical evidence for the utility of using tumor-infiltrating lymphocytes, those lymphocytes that naturally occur within metastatic melanoma or metastatic solid tumor. We believe that they may represent heterogeneous epitope recognition, so maybe they are the best selected cells for solid tumors. Of course, it has to be evaluated comparing to other cell therapies. So TILs or melanoma metastasis or solid tumor metastasis is being harvested, then the tumor-infiltrating lymphocytes are being cultured and within IL-2 medium, and they are being reinfused to the patient after preconditioning protocol of cyclophosphamide and fludarabine to induce lymphodepletion, and then the patient is treated with high-dose IL-2 in order to preserve TIL within the tumor tissue, within the blood system to induce a durable response. And according to the data that we knew, or if we look at one of our oldest patients from July 2009, you can see here peritoneal metastasis. This patient was previously treated with IL-2 and paclitaxel as part of clinical trial, and he had complete regression at the first assessment and ongoing until today. You see the CT from July 2013, but basically following 2 or 3 or 4 or 5 years, we end imaging tests. We don't repeat them anymore because most of these patients are cured. So this patient is cured from metastatic melanoma. And if we look on our previous data, on the data prior to the checkpoint inhibitors era, we can see that all these patients, looking at 103 patients, today we have enrolled over 200 patients, but these days we had these 8 patients that had complete response and all of them alive with no evidence of disease. We have some of the partial responders with very durable response and very durable survival. Some of the stable disease patients, but all of the progressive disease patients died because of metastatic melanoma. This is an example of another patient. And again, look at the year 2008, and he is regarded as partial responder only, not complete responder because he has residual lung lesions. Not necessarily these are metastases, but we didn't biopsy them to prove there are not live tumor cells within these lesions. So he is regarded as partial responder. And of course, those patients who have not complete resolution of all these imaging lesions, those evidenced lesions by CT or PET-CT, will be regarded as partial responders only, but the nature of the residual lesion is not clear, of course. What happened in melanoma and in oncology in general, the revolution of checkpoint inhibitors. And this slide from 2017 is a good one, which I continue to use. If you look at the curves, this is an overlay of seminar publications in melanoma, both targeted therapy and checkpoint inhibitors, the anti-PD-1, the anti-CTLA-4, and the combination of CTLA-4. And this curve at the bottom, the red one, reflects the results with ipilimumab, with anti-CTLA-4, which was the first checkpoint inhibitor available for use in metastatic melanoma. And even to recognize that this red curve shows the doubling of the survival of patients with metastatic melanoma, you can understand that the curves above this are a great achievement and significant change in the paradigm of treatment for patients with metastatic melanoma. What has it led us with TIL? To look again on the utility or the need for TIL treatment. Do we really need TIL for patients when we have off-the-shelf drugs to treat our patients, reaching up to 50-60% response rate in subset group of patients? Of course, we have huge unmet needs, those with high burden, high LDH disease at presentation, those with symptomatic brain metastasis, and some unusual melanomas like acral melanoma, mucosal melanoma, and conjunctival or choroidal melanoma, still a huge unmet need. But what about the regular cutaneous melanoma patient? We have checkpoint inhibitors that are available for use immediately.
TIL is a single treatment. It requires hospitalization for 2 or 3 weeks of the patients. Of course, it's expensive. It is a single patient, a patient-unique product. You can't produce generalized TIL for all the patients. You have to produce it for each patient in a GMP facility. And of course, some of the patients cannot finally get the compound, the TILs that we give them because they progress, because they have clinical deterioration, or because we can't, in very small subset of patients, we can produce the TIL. And of course, we have our donators, the Lemmelbaum family, but you need a huge economical support to hold this therapy for the patients. What happened with our patients? As you can see, looking at back there at 2017, 2018, 2020, it has affected the results of our treatment. We have been treating patients that failed checkpoint inhibitors, sometimes monotherapy, then combined immunotherapy, combined checkpoint inhibitors, sometimes also targeted therapy. And as you can see, if at the beginning, when we started at 2016, and then at 2006, 29, we had up to 40% response rate of our patients. When we reached those patients who were previously treated with targeted therapy, with immunotherapy, with checkpoint inhibitors, we got 0 response rate for our patients. So we got a much more resistant population and actually quite frustrating treatment for our patient. And actually, we enrolled quite few patients. We were unable to believe that we can affect significantly our patient with this demanding treatment. So questioning if we can move forward, if we should move forward, our answer was yes, because we still see 70% of the patients with metastatic melanoma treated with either immunotherapy, checkpoint inhibitors, or targeted therapy, still progress, still need advanced treatment for resistant, as a salvage therapy for resistant melanoma. And we need to have it with durable effect. But then we have to ask ourselves, do we choose the proper patient for treatment? What is the right time, the right patient to choose for adoptive cell transfer? Can we ensure that we are using the best cells, the product? Is it the best product that we have? Just harvesting the tumor? Should we harvest it early in the course of the disease, later just before we treat the patient? And there are some aspects for any decision that we take. Do we enable our cells to exert the maximal effect at the best terms, the preconditioning protocol, the high-dose IL-2, and of course, as always, IL-2, high-dose IL-2 is a highly toxic medication, and we have to ask ourselves if we have an alternative. So looking at data, we were not the only ones struggling with the new results of TIL therapy. This publication for 2021 from the NIH looked at patients that were previously treated with checkpoint inhibitor, comparing basically anti-PD-1 with those that were not previously exposed to anti-PD-1. Some data about anti-CTLA-4 shows that the response rate may be even increased in those patients that were previously treated with CTLA-4, and it may have an explanation. But as you can see, previously treated anti-PD-1 have much lower chance to respond or to have durable response to checkpoint blockade. Another publication from the NIH look at the subset of cells that we can identify, those patients that were treated with adoptive cell transfer. So if we look at the cell product that we look, and if we have those CD8 cells that express the exhaustion, terminally differentiated T cell, the CD39, the CD69 on the CD8, those patients are less likely to respond to adoptive cell transfer. Actually, their T cells showed terminally differentiation, much more exhaustion, and much less ability to attack their melanoma cells. So the question is, or actually this publication was from the era before the checkpoint inhibitors. Most of the patients that were tested for the expression of CD39, CD69 on their CD8 cells were patients that were treated previous to the checkpoint inhibitor era. So what we have to look, whether we have those patients pre-checkpoint inhibitors or post-checkpoint inhibitors, are those patients have more, the product, do we have more terminally exhausted T cells, terminally differentiated? Another question is if we can test this during time. So there is a small and nice publication looking at non-small cell lung patients for checkpoint inhibitor exposed patient. The tumor was biopsied pre-treatment, pre-checkpoint inhibitors treatment and post-checkpoint inhibitor treatment. And they were looking at resistance mechanism, immune therapy resistant mechanism. And they could see that although they were looking at a small number of patients, they could see various mechanisms of resistance. There isn't a single one. It's reasonable. Some of them indeed correlate with terminal differentiation of T cells. On the other hand, we had new publications in the last 2 years. Two publications that have altered our world again, changed our vision or the need to look again for T cells, adoptive cell transfer. One of them is a presentation or a publication in the JCO in 2021 on TILs, commercial TILs that are centrally produced for patients with metastatic melanoma. Theoretically, these patients were previously highly resistant to immunotherapy, and they could show about 36 response rate to adoptive cell transfer with these centrally produced TILs. So this publication, also some of them were durable response. We were looking and we were traveling to try to find and to discuss with our colleagues who are the patients that were enrolled to the study, if there are any differences in TIL production comparing to our TIL in our lab. Another publication from 2022 in the New England Journal of Medicine by the group of John Hannen from the Netherlands looked at TIL therapy at patients who failed checkpoint inhibitors, and for the first time they had a phase 3 trial with comparator arm of ipilimumab. So patients who failed anti-PD-1 were randomized to receive either adoptive cell transfer with TIL they produced in their center, locally produced, or ipilimumab. And again, they could show the benefit or the advantage of TIL therapy with response rate of 41% versus 18%, very different from our result comparing to ipilimumab. So, but looking deep into this publication, we asked ourselves, was it the real or was it the adequate comparator arm? Is ipilimumab is the right answer for patients who fail anti-PD-1? According to our clinician experience, naturally it is not. We know that there are hardly patients that can respond to ipilimumab following the failure of anti-PD-1. We can treat patients with the combination of anti-PD-1 and CTLA-4. This may yield up to 30% response rate, but monotherapy with anti-CTLA-4 is an inadequate choice. So the comparator arm is an issue. Also, looking at the patients that were enrolled, about 1/3 of the patients that were enrolled actually failed only adjuvant treatment with immunotherapy with anti-PD-1. So they weren't heavily pre-treated, fully exhausting immunotherapy. So looking again and bringing the clinician perspective and knowing that we have new studies and new data regarding new treatment option or available treatment option, we have to question ourselves whether the clinical trial was properly designed. Is the comparator arm is the proper one? Who are the patients that are being enrolled to the study? And what is the meaning? What is the true meaning? What is the true application of the results of the study? So, as I said, or as written at the bottom of the slide, one of the key probably is patient selection. Before terminal resistance has evolved, probably these are the patients that should be involved. And another example from a patient that was treated just recently by mid-2022, a young patient, she failed 4 prior lines of treatment, including targeted therapy and reinduction with targeted therapy. And if you look well, then there is a regression at the first assessment of breast lesion, breast metastasis, also lymph node metastasis, inguinal lymph node that has regressed. But this response, which wasn't durable, actually 3 months following the treatment, after a very quick regression of disease, she had new progression. She had new bone metastases that were not measured at the beginning, and she was symptomatic. And actually, it took about another 8 months before she died because of metastatic melanoma. So again, a patient that is truly highly pre-treated, very high pre-treated with 4 prior treatment lines, this is not the right patient to select for the study. But we are trying to have a better selection for our study. We try to pick those patients that didn't fail 2 years of immunotherapy, that received, progressed, and to pick them properly, are good in performance status. And if you can see, we have 2022, 2023 more responders among our patients. We don't have yet a good enough follow-up, a long enough follow-up to see the durability of response, but we know that we have to select patients more. We have more patients addressing or looking for adoptive cell transfer thanks to these 2 new publications, but we recognize the need to offer this demanding treatment to the best patients. So patient selection is one of the options. Looking at the TIL product, what we give to our patients, can we improve that? So one of our attempts was to try to look on those TILs that represent the CD137 or 41BB, which is an activation marker, meaning that if we can recognize those TILs among the TIL cell product and can enrich our TIL product with CD137 expressing TILs, then we may have more activated T cells. So we can see that we can increase interferon gamma secretion in cell culture if we have matched tumor, if we manage to grow the tumor for the patient and have an interferon ELISA secretion assay, and we can increase with these 41BB CD137 expressing cells. So we have, and we can also separate by specific beads the CD137 TILs from the total culture. So we have a group, a cohort of patients in our study that we can give CD137 enriched TILs for the patients. We are enrolling. If we manage to grow the TILs, we test for this expression and we can separate and enrich the TIL culture with these 41BB cells. Another direction that we look depends at another publication that we published in 2021, the study with fecal transplantation, microbiota transplantation. I won't repeat the results and the data of this study, and microbiome will be discussed later, but patients that were resistant to immune checkpoint inhibitors were treated with fecal microbiota transplantation for patients who are extreme or best responders to immunotherapy. So patients that responded for over a year were donators of stool, of microbiome, and patients were given those resistant patients, and 3 out of 10 patients responded to the treatment of checkpoint inhibitors following ingestion of responders' fecal microbiota. Another clue was biopsy that was taken to all of the patients from metastasis. So one of the metastases was selected for biopsy pre-treatment and post-treatment. And as you can see the infiltration of immune cells into the metastasis. And one of the mechanisms suggested is, of course, the recognition or eliciting more neoantigens by fecal microbiota, and probably by that activation, or one of the mechanisms to activate immune cells and to have an influx of immune cells into the tumor. Potentially, these may be effective TILs. So we have another cohort of patients in our study, in our TIL study. So we treat our patients with fecal microbiota just before harvesting the metastasis, several days, about a week before the harvest of metastasis. In this way, we try to increase antigen presentation and to have more TILs more effective against our melanoma cells. Another application of FMT is given about a week before hospitalization and before chemotherapy for the induction protocol. So the second patient is currently on treatment now, and we expect to enroll about 10 patients for this study. Another idea we are looking at in our lab is trying to reverse intracellular resistance mechanism. And this relies on a publication by Doug Palmer at 2022. They were looking on CISH, cytokine-induced SH2, an intracellular checkpoint. We know that expression of CISH drives an anergic phenotype of the T cells, and those T cells that have high CISH have also high exhaustion markers on their T cells, and that knocking out by CRISPR-Cis, we can increase interferon gamma secretion and increase cell killing by tumor-infiltrating lymphocytes. So their proposed clinical trial was to knock out or to offer knockout of the CISH after harvesting the TILs and growing the TIL culture. And indeed, there is a clinical trial now published at the clinicaltrial.gov with gastrointestinal cancer treated with tumor-infiltrating lymphocytes knocked by CISH. So we are looking in our lab now for TIL functionality, and you can see one knockout of both. We are looking not only for CISH, but for other molecules, trying to reactivate T cells by genetic manipulation. Two slides about other tumors. So we managed to grow TILs for other tumors as well, for ovarian cancer, for lung cancer, kidney, bladder cancer. The main thing to struggle, and we'll skip that, is an example with our ovarian cancer protocol. So we established and we opened a clinical trial for ovarian cancer patient about 2 years ago, and no patient was enrolled. And we are looking back at the inclusion-exclusion criteria. And again, looking at lab results and how we go to apply that for patients, we have to draw or to establish a clinical trial that is very wisely designed to check for inclusion-exclusion criteria to see if we are really able to enroll patients. For example, those patients with ovarian cancer, which may respond to chemotherapy, it's an issue to treat them with immunotherapy with having no evidence, and many struggles to enroll patients to this study. So just to summarize, we could see, and we still hope to see, TIL therapy with inducing durable effect for metastatic cancer. It's a single treatment, even though it's demanding, it's expensive as a single treatment. It's not comparing to a full year or 2 years or 4 years of checkpoint inhibitors. Not necessarily, it's not the winner in cost utility. It's an excellent platform to learn immune system and how to manipulate it. While checkpoint inhibitors became the backbone of cancer treatment, not only of melanoma, we have to consider how to give a good, effective salvage therapy. And maybe beyond melanoma, we have to address the question with TIL therapy. Do we have to choose cold tumors or hot tumors? How to manipulate those and properly design the clinical trials. I want to thank, this is some of the patients that are involved, but many other people are part of this effort to offer our patients a better salvage treatment.
Thank you, Ronnie, for giving us the clinical aspects. These are really important, and we're trying all the time to bridge the basic and the clinical research here. Just a request from our guests online. This is broadcasted live. Please, to the speakers, repeat the questions that come from the audience and speak louder. So, Asaf, please.
Naturally, as I said, there was a question about looking not only at the TIL product, but also at the microenvironment. Of course, this is a major issue, and we are aware of that. How we can affect that, as what I've tried to show is that moving forward with any idea means that you have to expand your clinical trial and to select the patient. But we are readily available or want and will be happy to collaborate in trying to think how to address this issue with another cohort of patients or to combine modalities, of course. So if I get it right, so you were asking about activation of the TILs extracorporeal after the harvest, if we can activate them better, except from IL-2. So great question. So we are looking, there are some offers or attempts to produce some other medium to produce the TILs, and also to treat the patient following the induction or the transfusion of TILs. We are part of a European consortium looking at attenuated IL-2, for example. But this will address mostly the toxicity of the IL-2. But basically, we grow our TILs in IL-2, and your question is perfectly right and need to be further investigated. Sorry.
Thank you, Ronnie. We'll have time during the break for more questions. All the speakers will stay here in the break. So to our next speaker, Professor Ravid Straussman from the Weizmann Institute of Science, who will speak about the tumor microbiome.
Okay. Hi, good morning, everyone, and thank you for inviting me to speak here today. This is more going back to the basic biology of cancer. If I want to summarize the talk in one sentence or two sentences, I would want to tell you that over the last 2-3 years, we found that fungi are also present in tumors. And we're trying to see that they are highly correlated to the response of patients to therapy, to the immune landscape of tumors, and we think this is going to be also an important new niche that need to be studied in cancer. So I'll show you all the rest now as details. I'll show you very shortly why do we think of looking into this and what the main findings. So we all recognize that tumors have a very complex microenvironment. And aside from cancer cells, you have immune cells and blood vessels and stromal cells. But over the last few years, our lab and other labs have been studying the microbiome of tumors, not of the gut, but inside tumors. And we're looking, for example, we started our journey in looking in pancreatic cancer, demonstrating here you see in LPS, staining in human pancreatic cancer, that there are LPS inside tumors. We could detect bacteria by FISH, by electron microscopy, and we demonstrated that it matters because patients that have bacteria in their tumors do not respond very well to treatment with gemcitabine because the bacteria can metabolize the drug, degrade it, inactivate it. And then Dvora and Ilana from my lab, together with Gary and Noam from the Open University and other members of the lab, something happened to the voice. We looked across different cancer types and asked, so are there also bacteria? You also hear this? Maybe someone can tune it down a bit, something happened to the volume. So try to look and ask, so are there bacteria in other cancer types as well? I can do like this. That's the best I can do. Okay, and very, very shortly, we demonstrated that bacteria present across different solid tumor types. We demonstrated that in each tumor type, there are different bacteria. We showed that the bacteria are usually present inside the cancer cells, as you can see here, for example, in breast cancer. And we demonstrated that these bacteria are mostly perinuclear. They're in very specific location inside the cancer cells. We're able to also to culture bacteria from tumors. And as Yardena also mentioned, together with her lab and Jen Wargo, we demonstrated that some bacterial peptides are presented on the MHC of cancer cells and can affect the tumor immune landscape. Going back to our story today, when I told you that we cultured bacteria from tumors, and from time to time we had, as you can see here, fungi that were cultured from the tumors. At first we thought this is probably a contamination because there was really almost nothing out there about fungi in tumors. But as we were getting more and more fungi over time, we thought, so is it possible that there are also fungi inside tumors? And as you know, everywhere in our world, when you have bacteria, you always have fungi. This is, for example, a screenshot from a Photoshop from a gut of mice in this case, but this is true for human as well. And you can see the bacteria, of course, in red and many fungi in our gut. And this is true for our skin and everywhere else. And fungi is really an amazing kingdom. People think there is between 2 and 4 million species. In the fungal kingdom, only around 150,000 of them we know, and around 300 of them are known to be pathogenic to patients. But there's much more to learn about this amazing kingdom. And Liane, which was a PhD student in my lab, decided to look into this. So she took tumor samples from more than 100 patients. And when she looked at the fungal specific 5.8S by qPCR, she did find fungal DNA inside tumors. So we officially declared this as a project in the lab and decided to look into it and try to find out the really fungi inside tumors. And just to say that this was done with a very tight collaboration with Professor Satchi-Fainaro from the Weizmann, Lian and Ilana from my lab led this project, and also collaborating with Rob Knight and his PhD student Greg Poor, and close collaboration with Amir Bashan and his student Guy Amit from Bar-Ilan University, and also many other collaborators around the world that helped us in this project. So overall, we characterized more than 17,000 tumors from patients from 35 different cancer types, and we had hundreds of negative control to make sure that what we characterize are really intra-tumor fungi, not just contaminations coming in the way. Published it around a year back, and we stained many of these tumors to see the fungi. We sequenced the ITS2 to find out which fungi are there, and we also used TCGA data to analyze reads, try to filter out human reads and see if we find fungal reads there. So what did we find? So in staining, if you actually look for fungi, you can definitely find them inside tumors. So here's for example, in the ovarian cancer, you can see a budding yeast inside this patient tumor, and when you sequence the tumor, you see a known yeast. And in this lung cancer, we found something that looked like aspergillus with GMS staining. It was confirmed with fish staining, and again, when we sequenced this tumor, we found aspergillus. And we found that just like in bacteria, mostly we see the fungi intracellular and not between the cells. So here you can see, and we found them in immune and cancer cells. Here you can see a melanoma example. You see positive staining for a beta-glucan, fungal beta-glucan. It's inside macrophages, and specifically we find aspergillus in this patient inside this macrophages CD4, D5, CD68 positive cells. But in this pancreatic cancer, for example, we find the alpha beta-glucan staining inside the cancer cell, only in the cancer cells, as you can see here, confirmed by fish. But you can see in the H&E that this is really the epithelial compartment. These are the cancer cells, negative for immune cells. And we're definitely not the first one demonstrating that fungi can invade the intracellular location. So maybe not in cancer, but there's many publications. Here's, for example, Candida albicans, and here's aspergillus. So people have found these fungi inside cancer cells. So we wanted to do a really thorough investigation and ask which fungi are present inside human tumors. So as I said, we looked at TCGA data, more than 15,000 samples, as well as almost 1,200 samples that we sequenced in the lab. And I want to just say that we have hundreds of negative control, as I said, to make sure we're really profiling what's in the tumor, but it's very different from bacteria. So when we do the same for bacteria, we usually have to filter out 95% of the bacteria that we found because we have a lot of indication that they're coming from contamination. But so for some reason, in the fungal world, we don't see it. Most of the fungi that we see are coming from the tumors, and it seems that there's much less fungal DNA in the kits that we're using, in the operating room, etc. So most of what we find is actually coming from the tumor. And non-surprisingly, we found mostly fungi belonging to the Basidiomycota and Ascomycota phyla. And it was nice to see, for example, that in melanoma, the skin cancer, we mostly find Malassezia which is the most common fungi of the skin. And in colon, we find Saccharomycetes, as one can guess. And again, we find that there's different fungal species in different tumor types. So if you cluster them, you find, for example, Candida albicans and Saccharomycetes in colon. We find other fungi in breast and so on. And if you look at the tissue that is adjacent to the tumor, you kind of find the same fungi, which is not to say if these are present in normal tissue or not, but in the tissue that surrounds the tumor, we can also find these fungi. So how many fungi can you find inside tumors? If you look at the richness, you say how many types of fungi, the answer is extremely low numbers. So usually we find either zero, some patients have no fungi in their tumors, or we find usually one or two. And breast and bone are uniquely have a rich microbiome, and there's a very tight correlation with the bacteria inside tumors. So mostly we see that patients that have a lot of bacteria in the tumors tend to have more fungi in their tumors and vice versa. And our hypothesis is that this really just reflects the immune system in the tumor. So patients with a more suppressed immune system would have probably more bacteria and more fungi, and vice versa. When you look at the TCGA data, you can see that you can find fungal reads and bacterial reads across all different tumor types, but mostly we have less fungal reads than bacterial reads. So we would calculate there's at least 10 times less fungi than bacteria in tumors. And then one can ask, just as people are taking liquid biopsies and asking, can we detect tumor DNA in the blood? You can ask, can we detect this unique tumor bacterial and fungal DNA in the blood? And can we differentiate cancer cells, cancer patients from normal? And the answer is yes. Just by amplifying, by sequencing bacterial and fungal DNA, if you look at multi-domain features, so all bacteria, viruses, and fungal, you can actually very accurately differentiate between healthy and patients with cancer. I also say that we also already got FDA approval for a single test for patients that have a lesion that was found in imaging in their lung, and it's not sure if this is cancer or not. And usually the next step is to take a biopsy. But of course, there is complication rate of such biopsies. And now it's been approved to take the blood test, look for fungal and bacterial DNA, and then segregate the patients into those that have a low risk and they can wait a year and see what happens, and those that have a high risk that should proceed to do a proper biopsy. I can also say that we found very strong correlations with clinical data of the patients. I'll just give you a few examples. In 2020, we showed that, for example, in lung tumors from smokers, we can find more bacteria than non-smokers. And here we see the same. If you take lung tumors from smokers, we find that the richness of fungi is higher, and there's specific fungi, for example, here Aspergillus, that almost exclusively found in smokers and cannot find in non-smokers. We also find correlations with overall survival. Here, for example, you see a small cohort of 80 breast cancer patients, but you can see that those patients that have malassezia globosa in their tumors has a much more poor prognosis than those that do not have fungi. And again, this might just indicate the well-being of the immune system inside these tumors. As was mentioned here, and the gut microbiome is tightly correlated with response to immunotherapy. We also find some specific fungi inside the tumor that are correlated with response to immunotherapy. And also I want to say that we did a quite thorough study to look at the network between fungi and bacteria inside tumors. And I think surprisingly, we find that mostly it's a positive correlation. So the red lines are the negative one, but all the rest, all the gray ones are positive correlations. So there's some hubs, like a malassezia hub and Aspergillus hub, that if you have these fungi, you tend to have other specific bacteria in your tumors, but not just bacteria, also the immune system. So here you can see a summary of more than 15,000 patients that we have their fungal characterization of the tumors, the bacteria, and also the immune cells. And by clustering it, we found that there's three main, what we call mycotypes. So some patients have a malassezia mycotype, and this is correlated with specific bacteria and specific immune cells. And others have a more Aspergillus candida mycotype, and this seems to be correlated with other bacteria and other immune cells. And lastly, we have a third group that have different types of fungi that correlate with completely different bacteria and different immune cells. And I would just say in one sentence that now we have a very large project in the lab in which we try to understand how these bacteria and fungi affect the local immune system. We're doing it by profiling more than 300 patients for which we know their fungal and bacterial composition, the response to immunotherapy, but also transcriptomics and immunohistochemistry. And we do find that there's both bacterial and fungal taxa that are highly associated with response, but I'll talk about it some other time. So to summarize, there seems to be different tumor types have different fungal profiles. We know that most patients have a very low richness of a microbiome. Most fungi that we detect are intracellular. We see them both in the tumor and the normal adjacent tissue. we see very striking fungal bacterial immune co-occurrences that we are studying. And we know that many fungi, their presence, both the total load of fungi as well as specific fungi are highly correlated with immune response. Okay, so I'm missing a slide here. I think there was another slide, but I just want to say two things. First of all, that this was done by many people that helped us. I can't go over all of them, so I want to thank them very much. This is my lab, and I want to thank mostly Lian and Ilana that I showed you their results today. But I also want to say another thing, that we have an unrelated lecture. We have a really nice novel initiative with the Weizmann. We're doing the first cancer hackathon, we call it. This is going to be on 31 of March, so in 2 months from now. It's a really unique opportunity. It's going to be centered around 2 metastatic breast cancer patients. And the idea is that every year in this hackathon, we're going to really push the boundaries and as far as we can, but doing it around real patients. And every year there's going to be a specific idea or a specific area of research. And this year is going to be personalized immunotherapy. So I don't mean precision immunotherapy, not trying to match the right drug to the right patients, but actually, if you have specific patients and you have a lot of resources, can you really design the best therapy for these patients? TILs would be one example for that, but there's many others. So we're going to have specific antibodies or vaccines or TILs or TCRs or CAR T cells. And there's going to be a website probably later this week or early next week. But if someone is interested to come and not only participate, but also help in this hackathon, please feel free to reach me by email and I'll be happy to give all the details. תודה רבה. Yes. So yeah. So as for whether they're alive, first of all, we started seeing these by culturing them from tumors. This is a good indication that at least some of what we see are alive. I truly agree that maybe some of the staining that we see, or more I would say the DNA, might be some fungi or bacteria inside macrophages, but we do see them when we culture them, and also we do some live staining. We can take 2 tissues, for example, in EVOC, ex vivo organ culture, and we can stain them with specific things that can only integrate into the bacteria or fungi when they are alive, and we definitely see them. So I would say that some of them at least are alive. And as for the entry, I agree that I would guess that mostly are coming from the blood. It can be carried inside macrophages. You say like this, why? Because I mean, we know that there are bacteria in our blood every day, probably also fungi, just like we have transient bacteremias. You might have some bacteria and some fungi coming through the bloodstream. If you're healthy, the immune system will take care of that, right? But if you have a unique niche, like a tumor that is immune suppressed and has a fenestrated vasculature, you can think that these bacteria and fungi will reach the tumor through our bloodstream. So that would be my guess. I would be very careful with that because, when you give antibiotics, antifungal, the effects are going to be systemic. As we know, for example, for antibiotics, when you use antibiotics of patients actually have the opposite effect. It can harm the response to immunotherapy. So I think we should be very careful. What we're trying to do in the lab is not to modulate now the bacteria and fungi by antibiotics, but actually to understand the basic mechanisms. What are the specific bacterial and fungal genes that correlate with response and non-response? And then we're thinking about targeted therapies to these specific functions. I think that modulating the whole microbiome might be too crude of a way of dealing with this issue.
So we'll have time for sure. 2 short questions and 2 very short answers.
Okay.
Trying to accommodate. Shai.
Answer, we don't know enough. Next. That was short enough?
Yes.
Very shortly, I would say that in most organs, we don't know because we don't have true normal. Usually what we have is normal adjacent to tumor, and in these areas we do have fungi and bacteria. I would say the only tissue we have true normal is breast cancer for patients that had breast reduction surgeries for non-cancer reasons, and we do see both fungi and bacteria in these tissues. Yeah, so that's exactly the paper that we published with Yardena in Nature 2021, which they found specific TCRs for bacterial peptides in melanoma.
Thank you.
Thank you. This was really fascinating. I have tons of questions for the break for you, but we'll wait with that. So our next speaker is Professor Shai Izraeli from the Schneider Children's Hospital and Tel Aviv University.
So I deliberately asked these questions about the blood because we're moving to blood, right? So I'm a physician scientist, a true kind of physician scientist, meaning I have a split mind and culture, etc. But that also means for our research that every research that we have been doing in our lab started with patients. And the usual course is that you identify a problem, an unmet need, a curiosity in a patient, you take it to a lab, it can be your lab or somebody else's lab, and then you find all kind of discoveries, you make discoveries that may often not related to the specific patients you started with, but that are important and they return eventually to patients. So this is the ideal translational research cycle. And people who know our research know that most of our research over the last 23 years has been started because of one patient that I've seen in the clinic, and this is a Down patient with leukemia. And then I learned that Down syndrome is a very high risk syndrome for leukemia. And indeed, we made a lot of research in Down syndrome that led to many discoveries that are unrelated to Down syndrome. The most recent one is a way to treat high-risk leukemia. This was just published in Nature Communication, and go look at it because I'm not going to talk about this. Instead, I'm going to talk about this very special patient to me, because this is my late father, who died already in 1993, so this is 30 years ago. And for some time, the last 2 months, I distributed a little speech that I will not repeat here, about how happy I am that he has not lived on 7th of October, because on October 7, he would experience a replay of what happened to him during his childhood in the Nazi time. But we are not talking about this now. On his lap is a baby. This is my son. During his, so this is the brief. And 30 years ago, this son is a doctor now. And I show this, and I say a very special patient, because at that time he was dying, my father, of course, not my son, from lung cancer brain metastasis. And since then, I couldn't stop thinking why lung cancer cells go to the brain. Lung cancer and melanoma are the two most common reason for brain metastasis. More recently, breast cancer became so because of the activity of brain cancer, of therapy to breast cancer. And this guy, Stephen Paget, many years ago, noticed also the phenomena. I mean, they were really clinicians, and he phrased the seed and soil hypothesis. And this hypothesis was that cancer metastasis, cancer cells, are like seeds, and they need a fertile soil. So probably the brain produced something that encourage lung cancer cell to grow them. And since then we published a couple of papers on lung cancer cell metastasis. the most recent one in collaboration with Yair Bar. But as I told you in the beginning, my interest is in leukemia. It's actually great to be, this is the International Cancer Day, and it's fantastic to be in this day because childhood leukemia is one of the celebrations that we rightfully celebrate. So this is the prognosis of childhood leukemia when I was a child, 6-day war, zero patients were cured. This is when I was a student, this is when I was intern, and this is now when I'm a department chair. So you see, and everything is because of me, you see, child, you know. Now, one of the major jumps in the cure of childhood leukemia was in the 70s, early 80s, when CNS prophylaxis was introduced. So let's talk about it. So when you know, in the 60s, prednisone, vincristine, methotrexate were introduced, and suddenly people could see complete remission in leukemias. But then the problem that most of the patients recurred in the CNS. The clinician then hypothesized that at the time of presentation, there are already cells in the CNS that we cannot see. So if we give prophylaxis to the CNS, then maybe we can cure leukemia. And that apparently was the case, and the prophylaxis was radiation, quite hefty dose of radiation, 204,000 rad, or 2.4 gray, as it is used to be told today. And this was amazing, really amazing. And I had the opportunity when we still flew, this was before October 7, when we still flew, to have someone sitting near me. You know, I talked with her, and she was one of the first patients in Israel in the 80s to be cured thanks to this CNS prophylaxis by radiation. So that's the good part of it. So now we have only about 5% or 3% CNS leukemia, but in an era where we have only about 5% relapses, they constitute 30 to 50% of the relapses. So CNS relapses are really hard to treat. But what the flip-flop of this incredible therapy was incredible toxicity. So the two types of toxicities, very serious neurocognitive defects. So this type of radiation led to about 10 points, not 10%. Yeah, they're actually percent. 10 points decrease in IQ and secondary brain tumors. And therefore, actually the Israeli group led by the late Professor Rina Zaitsov was the first in the world that omitted CNS prophylaxis and changed it with intrathecal chemotherapy. And recently this has become the standard, and it's almost CNS prophylaxis by radiation is almost never given today. But again, this is given already for a long time to realize, and really you don't need to be a scientist to understand that giving chemotherapy to a brain of a child or a 4 or 5 or 6-year-old is not really a great idea. So they are milder, much milder neurocognitive defects, and that's the status today. So, and you see this in these two papers. So, you know, so that's a really good reason to study CNS leukemia. So not only my father story, but other patient stories. And of course, we have to go to the basic and to ask the question, which cells enter the CNS, route of entry, is the two-way traffic. Can cells from the CNS can go back and do a systemic relapse? Why they go there? What is the seed-soil relationship? And potential novel cures. Now I have only about 10 plus minutes, so of course I cannot talk about all these points. I only will mention a couple of really interesting things that came from the literature and then talk about our work. So what is really exciting is a novel discovery. It's funny that you can still make discoveries in anatomy today after all these little microbacteria inside tumors. So it has been discovered that the same route that is taken by to developing neurons from the periphery into the brain is also for leukemic cells. So they stick two blood vessels going from the bone marrow in the bone here into the brain, and they use a additional molecule that is called alpha-6 integrin, and I mentioned this because now there are antibodies against alpha-6 integrins in clinical trials. So this may be useful for prevention of more CNS leukemia, but this will have no activity against the cells that are already there, and that we don't know about them because there are too few to detect. Again, this anatomy discoveries only very recently. Jonathan Kipnis, who is an Israeli that sits in UVA, discovered in Nature a couple of years ago a whole lymphatic system that was unknown, that cells can move from the brain into the body. So now maybe these cells are not only sitting there, but maybe also infecting the body. But what we are interested in the question, could CNS leukemia, high risk for CNS leukemia could be identified, and those for low risk. So we can give toxic therapy only to those patients who are high risk, and maybe also we can find specific therapies. So we were interested in this. We are interested in these questions for I don't know, at least 10 years. And I'm looking here at Angel Porgador because we collaborated in this. You'll have to read if you're interested in a study that shows the importance of interleukin 15 and NK cells in modulating CNS leukemia. These studies were confirmed by others. But today I want to tell you about a much more recent study that was led by Angie Maria Savino, who has now her own lab in Italy. And she hypothesized somewhat the opposite of the famous Paget hypothesis. She said maybe it's not the seed, the soil that fits the seed. Maybe the seed can go adaptation to this soil. And to check this hypothesis, we did a rather simple experiment. This was actually already done before Angie by Liron Frischman. And what she did, she injected leukemic cells human leukemic cells into immune deficient mice. And you know, they always colonize the CNS. She took cells from the CNS, spleen, bone marrow, did gene expression studies and see what pathways distinguish them. And we were expected all kind of pathways. But what we found, we never expected. That's why they say if you don't know what to do, you do genomics. But genomics is something worthwhile because what we found was completely unexpected. What we found is that the pathway that was most upregulated in the cells that infiltrated the brain was the synthetic fatty acid pathway. Now, fatty acids are a source of energy, but here they are not used for energy. Synthetic of fatty acid. Why do you need to synthesize fatty acids? We verified this by RNA, protein on human cells, and the most critical enzyme is an enzyme called SCD. I cannot pronounce this theoryl coenzyme SCD. And this SCD is the entrance to fatty acid synthesis. Why we need fatty acid synthesis? For membranes in signal transduction. So lo and behold, we showed that the CSF is completely empty of fatty acids. It's a desert. Cells cannot live without fatty acids. We showed that if we express SCD in cells, they will go much more to the CNS. And we showed that SCD is a target for therapy. So if you knock it out, cells disappear in the CNS. If you block it, cells disappear in the CNS. And it's true also for primary material. So to summarize these parts, we found that there is a lake of unsaturated fatty acid in the CSF. Leukemia needs to upregulate this pathway, and it's a target for therapy. But it's almost the end. It's not the end, it's almost the end, because just after we publish, in 2 papers already citing us, showing exactly the same for breast cancer. So this is biology. It's not leukemia or cancer. In order for cancer cells to survive in the CNS, they need to synthesize fatty acid. So first, it's important because now companies are interested in making inhibitors because this is a big market. And the second is this change in the hypothesis of Mr. Paget from seed and soil adaptation to the soil hypothesis of cancer metastasis. Thank you very much. This is the advertisement, and these are all the collaborators. Thank you.
Thank you, Shai, for a wonderful talk. Dino?
So I'll repeat this question. What Dino asked is what stimulate? Is this the extracellular matrix, for example? There is a simple way to test it, by the way, to grow them in CSF and to see what happens. We didn't do the experiment, although it's simple. It's interesting because, you know, in the CSF, the cells are not floating. It has been shown that if they float, they die. They stick to one each other. So maybe this has something to do. I'm just thinking while talking, maybe it's this cell-to-cell interaction, because they really form a solid tumor within the CSF. But I don't know the answer. Well, Yuval asked about side effects. That's also always an issue. You know, there is no drug, not even a single drug. This is no side effects, right? And all targeted therapy has side effects because they block physiological. So it's always a question of therapeutic ratio. Having said that, I think that SCD inhibitors were gone into clinical trials, and the 2 major effects that killed them were acne. Acne is something that kills drug, and fatty liver. But now they are talking about developing inhibitors of not of SCD but of enzyme in the chain. So you can imagine all kind of things, and also because I got marked that I'm going over my time. I didn't show you this. I saw this slide, but I didn't. But I'm stealing the time now. It's interesting, very interesting. When we knocked out the gene, the cells didn't grow in the brain, but they grow much better in the bone marrow. Why? They got fat from the fatty cells. When we inhibited pharmacologically, they didn't grow neither in the bone marrow nor in the brain. So that's the difference between pharmacology and genetics. So the question, what will be the effect on the microenvironment of inhibitors? I think I just gave an example. The fatty acids themselves, I don't know, but the inhibitors, I just gave an example that they block the growth of leukemic cells in the bone marrow because probably they block the production of fatty acid by microenvironment. But this is a whole new field, and actually there are lots of papers coming on this field of various aspects of lipidomic, actually. It's a very interesting field. And I must mention Eyal Gottlieb for his contribution also, and his specific interest in lipidomics.
Thank you, Shai. Last but not least, before the break, our next speaker is Professor Angel Porgador from Ben-Gurion University of the Negev. We'll speak about artificial reporters for personalized cancer immunotherapy.
So thank you, Ronit, for inviting me to this respected meeting. And I understand it's worldwide broadcasting, so luckily I bought my jacket. Okay, so it's about artificial reporters for personalized cancer immunotherapy. So it's only 20 minutes, so I will just talk about indeed this subject here. And this project was done in full collaboration with Professor Moshe Alkabes from Ben-Gurion University, MD PhD student Bar Kaufman, and first year PhD student Orly Abramov. So is cancer immunotherapy important? We can see actually from Google Trends that the interest of the public in cancer immunotherapy surged from 2013. I think it was the time that the first immune checkpoint inhibitor was published on melanoma. And it's but the one interesting thing is we can see here from the comparison of the word search for cancer immunotherapy and pembrolizumab that most of the interest was actually around immune checkpoint blockers because it goes hand by hand. So definitely immune check blockers, this is one of the two major advancements in cancer immunotherapy in addition to ACT, to CAR therapy. And the basis is very simple. So we have it was first published by James Allison back in 1995 that in addition to first signal, the TCR recognition, co-stimulation signal, there is also inhibitory signal. He was describing the CTLA4 and then the PD-1 was described. And basically, if you have recognition of the T cell, of the tumor cell, and this is the first signal, the TCR-based activation. You have also inhibitory signal between PD-1 receptor on the T cells and PD-L1 expressed on the membrane of the tumor cells, or antigen-presenting cells in the tumor microenvironment. But what we was shown, the plus and minus actually make usually minus for the T cell, but then the development of the immune checkpoint blockers, mostly monoclonal antibodies, you can block either the PD-1 by antibody to PD-1 or the PD-L1 by antibody to PD-L1, and then you will are staying only with the plus, and the T cells can actually try to kill or eliminate the tumor cells and induce death of the tumor cells. So that's great. And back in 2020, 43% of US patients with cancer were, today more, eligible for immune checkpoint blockade or immune checkpoint inhibition therapy, but only 12% of the 43% responded, so about 5%. So comes back to the question of personalized cancer immunotherapy. How can we match the treatment to the ones that will clinically responded? Ravid was mentioning the difference between personalized and precision cancer medicine. To my point of view, personalized is the matching, precision when it actually works. So, and that's the question, how can we contribute to the fact that we really match it, that it will work, not just match it to the patient? And so, naturally, the first idea of the people who try to match between patients that will respond to immune checkpoint blocking was, let's stain for the density, using immunohistochemistry of the ligand to the immune checkpoint receptor on the tumor cells or in the tumor microenvironment, on the antigen-presenting cells also. But that's actually so PD-1 is the one that was mostly studied, and PD-L1 is one of the two antigens or ligands to PD-1. And a lot of studies were done with PD-L1 staining, which is actually a clinical protocol to define, at least was before, now less, who is eligible for PD-1 therapy. And the problem was that PD-L1 staining with immunohistochemistry failed to accurately predict the clinical outcome with treatment with PD-1. It's like a dismal area under the curve of 0.55 to 0.60, a little bit more than just flipping a coin. So what can we do? So we hypothesize that it's not the density as measured by antibodies of the ligand, it's the functional bioavailability of the immune checkpoint ligands that can predict the response of the patients to treatment with anti-PD-1 therapy. So based on this hypothesis, we decided to develop immune checkpoint artificial reporters that we can use them to quantify the functional bioavailability of the ligands to the receptor. So, and we already published that a few months ago, but that's what I'm going to discuss briefly now. So the first question is, why functional bioavailability and not, why it would be better than staining for the density of the ligands on the cells, on the tumor cells, or in the tumor microenvironment. So there are several reasons. I just named some of them. First, and for sure, it's the presence of multiple ligands to the immune checkpoint receptor. Actually, the PD-1 is a simple case. You have only PD-L1 and PD-L2. Other checkpoints that will come to the market, already came, will come in the coming few years, have much more ligands, and some of them are much harder to quantify because they are based on glycosylation or glycans. So, and in the clinical now, nobody stains for PD-L2, only for PD-L1, but there is PD-L2 is actually 3 times have higher affinity to the PD-1, but nobody studied it clinically because PD-L1 was the first one to be defined, so everybody went for the PD-L1. So then you have the glycosylation of the immune. All of these ligands are actually glycoproteins, PD-L1, PD-L2. They have glycosylation, tumor alter glycosylation. The changes of the glycosylation are usually not reflected by the binding of artificial monoclonal antibodies developed against the ligands, but it's definitely reflecting on the affinity of the PD-1 receptor. So you might have some kind of difference between staining an antibody and trying to define it with the receptor. Then, of course, you have the life cycle of the ligand. What is the balance between the staining or the presence of the ligand in vivo, within the cell, intra the cell, and on the membrane. So when you stain, actually, immunohistochemistry, you stain for everything. When you come with the functional receptor to try to see whether it works or not, you actually go for the membrane staining, for the membrane expression. And of course, there is the issue of cis-trans inhibition of the ligands with other ligands, and there is a very known case for the expression cis or trans of PD-L1 and CD80 coming from another immune checkpoint pathway, the CTLA4, that can inhibit either the CTLA4 or the PD-1 pathways. And of course, there is the membrane distribution aggregation on the membrane, which affect the way that the receptor is activated, but doesn't change the staining with an antibody. So this is just some of the options that can explain the difference between trying to understand whether the patient will respond to anti-PD-1 therapy using staining in immunohistochemistry with artificial monoclonal antibodies or trying to use the receptor. But how can we use actually the receptor? Basically by trying to imitate the T cell. So the T cell is here, the T cell has PD-1, of course, has also the TCR, recognizing either PD-L1 or PD-L2, and then you can come with antibody that block the PD-1 or clinically antibody that block the PD-L2 to try to enhance the activity of the T cell. There is no clinical antibody to PD-L2, of course, unfortunately, and this could be the T cell can recognize either cancer cells or antigen-presenting cells in the tumor microenvironment. So what we developed is the immune checkpoint artificial reporter, in short ICAR, in which in that case we replace the native T cell or the patient T cells with an artificial reporter based on murine thymoma BW51447, and in which to this reporter we introduce the PD-1 receptor, the human PD-1 receptor, fused to murine zeta. So any interaction of this PD-1 with the functional ligand will induce IL-2, murine IL-2 secretion for the reporter. And murine IL-2 is a great marker, even though everybody likes to use a fluorescent marker because it has a very high range of response. So you can actually understand your limitations or the limitations of the system. And of course, the other thing that you can do, you can block now the recombinant PD-1 with the clinical drugs, either antibody to PD-1 or antibody to PD-L1 on the target tumor cells. Remember, there are target tumor cells here, and I will show from where. And then you will get reduction, hopefully, in the IL-2 secretion, and you can combine all of that, the intensity and the reduction following treatment with the drug and get some kind of a score. I'll show it later. So that's the reporter, and the first thing that we did after developing the reporter and studying it and optimizing it, of course, we selected some cell lines. We enhanced the expression of PD-L1, PD-L2 on that, and for most cell lines, the ones that are actually fitting the regular dogma and not the ones that are answering some of the issues that I mentioned before. We saw a great, very nice correlation between the ICAR response. So this is ICAR with recombinant PD-1, secreting IL-2 following exposure to PD-L1 or PD-L2 present on the target cells. So we saw a very nice correlation between the PD-L1 staining using flow cytometry or PD-L2 staining where it was appropriate. to the ICAR response. But cell lines are not the case with patients. The case with patients, what you have is basically you have a block. In the best case, you have a block, fixed tumor tissue, either from a biopsy or resection, and you need to make your decision based on cuts from this block. So that's actually faces a real challenge, because when you are thinking functional assay with reporter, you naturally do not think that fixed dead tissue will be involved as target to a functional assay with the reporter, but that was not the case. We employed the patient-derived xenograph system that we have in actually to generate from the same PDX grown in NSG mice, either fresh cells that we could test them with the reporter, or blocked fixed cells. We generate the block here using exactly the same clinical pathology lab, and then we cut some 5-micron cuts from the block, antigen revive them. So this is not making the cells alive again. We are not. What was the name of the physician that actually created Frankenstein? So we are not there. We didn't revive the cells, but we did revive the structure and the distribution of the ligands on the fixed cells using antigen retrieval. Very meticulously searched to find the optimal one, and you compared the both results, and we're happy and even surprised to see that there is a complete correlation between the response of the ICAR PD-1 reporter to fresh tissue as compared to fixed antigen retrieved tissue. Of course, it's not on the same scale, but it's completely correlated. So now when we had these results, we were freely open to do some kind of a retrospective study using our reporter and basically getting blocks from patients that were already treated with anti-PD-1 monotherapy. The clinical results were already known, but now we could get 4 to 5 microns, no need for dry ice, nothing, just by an Eppendorf, 4 to 5 cuts of 5 micron with each, getting them, doing the antigen retrieval, putting them, plating them on the 96-48 well plates, and basically ask whether we can see a response from the reporter and what happened when we add the clinical drug. It could be anti-PD-1, so there is a score for anti-PD-1, but it's also we can add, instead of anti-PD-1, we can add anti-PD-L1, which recognize the tumor cells, the revived tumor cells, and get a score for anti-PD-L1 and even for anti-PD-L2 using a commercial blocking antibody to anti-PD-L2. So in this study of over 70 samples, retrospective samples, we had samples from Barzilai, Soroka, Rambam, different histotypes, head and neck, non-small lung cancer, TCC, RCC, and others. And basically, so we actually got this after quite few months works. We got that the ICAR PD-1 score, let me explain what do I mean by ICAR PD-1 score. So we take the reporter, we take the revived tumor structure cells from the block, we put them together, we get intensity, which is the IL-2 secreted, should there is a functional PD-L1 or PD-L2 on the tumor cells. Then we add the clinical drug, and we should see if there is an intensity of response, we should see reduction. If it works, it might be complete or partial reduction, and the coverage of the slide and some positive and negative standards in order that we can compare results from different time points of doing the studies, we get a score. So this is the formula. And I just mentioned again, this is a formula that is based, the score is based on using a specific clinical drug. Here you see a graph with the score results using anti-PD-1 clinical drug, the pembrolizumab. We can do it also for nivolumab or cemiplimab. And we were surprised to see, actually surprised, that the difference between non-clinically responders and our two responders, our score is high when you are expected to be responding, is very clear and very nice. Now we are repeating this in a proof of concept with a commercial company, and very nice results already. So this is the results. This is the main slide of this presentation. We can actually come also to analysis of the survival of the patients using the 4 value of the score as a differentiating for this 70 and more samples, and this is the differences in survival. When the score is above 4, much significantly, score of the hours assay, much more survivors, as you can see the blue line here compared to here. And this is, of course, the staining, the PD-L1 regular staining below 1%, above 1%, and there is no, as I mentioned, there is not effect or there is no prediction capacity, good prediction capacity to PD-L1 staining in immunohistochemistry. So we believe that using ICAR PD-1 to identify responders can actually help to identify the responders. But now we're beginning to understand the reason that I mentioned at the beginning, what is the difference between staining with the antibody and doing functional assay. And one of the major differences is the glycosylation of the ligands. Again, something that is not reflected well by the artificial antibodies produced either to stain or treat patients. But here we collaborated with Adit Ben-Baruch, Professor Adit Ben-Baruch from Tel Aviv University, and with Nofar Erlichman and Tzipi Meshel from the lab of Adit. And they actually studied non-immunogenic roles of PD-L1 and generated a series of PD-L1s on 2 models, MDA and FCF, that are lacking one or all of the glycosylation, the 4 N-glycosylation sites of PD-L1, here in position 35 and here in position 192, 200, and 219. Interaction with the PD-1 is through the membrane distal domain. This is PD-L1 here. And we got something that actually surprised us very much. We expected that what we'll see with these glycosylation mutants effect on the anti-PD-L1 score. But what we mostly got was effect on the anti-PD-1 score. And why it is surprising? Because our reporter expressed one type of PD-1, recombinant PD-1. So why different glycosylation on the PD-L1 target will affect the anti-PD-1, but that basically blocks the PD-1, not the PD-L1. But here we got something amazing. For example, and you can see it mostly for the cemiplimab. Again, remember, you have anti-PD-1 clinical drugs. Believe me, they are not the same in efficiency. It's known, but there is no way to compare it per each patient. So for the cemiplimab, you see that if we have a mutant which doesn't have any glycosylation, you can block very easily our reporter. Very low concentration, all activity of the reporter is blocked with the cemiplimab. However, when you have the N35 mutation, meaning that the PD-L1 expressed by the MDA or the MCF cancer cell line only is missing the mutation in N35 in the membrane distal domain, so suddenly cemiplimab cannot block, even though it binds to the PD-1 on our reporter. So there are explanations to this. I can assure that we are doing modeling. It's connected to the life cycle on the membrane. It's connected to the secretion, to the supernatant. So there are explanations. We are working on that, but this is already a basic study. I want to finish it by saying that today, basically, most of the decisions of the physicians are based on the CTLA-4 and the PD-1 pathways, whether to give immune checkpoint or not, but there are so many other inhibitory molecules that affect the way that the T cells recognize and kill the tumor cells. So, and of course, a lot of drugs are now developed, and some of them already, the anti-LAG-3 already came to the market, the anti-CTLA-4 is already there, but it's the situation today, not in the coming few years. It reminds me the question of the famous question in Hamlet is, to be or not to be, that is the question. We can replace it with that. To give or not to give PD-1, PD-L1 based immunocheck, that's actually the decision of the oncologist. Should they give anti-PD-1 or shouldn't they give anti-PD-1? But that's not the case in the coming few years, because you will have much more options what to give. You will have you have already several immune checkpoint blockers per each immune checkpoint pipeline, and I can assure you it's not the same efficiency. You will have new approved ICBs for different IC pathways, the LAG-3, the TIM-3, the BTLA, the TIGIT, so you have much more. And you definitely will have the era of ICB combination, because it is clear today that to reach to much more than 12% response, average between different cancers, to ICB, you have to combine 2 ICBs at least, and what is the right combinations. So having the ligand density studies in IHC will not help. As I said, the PD-L1 that didn't work was the easiest case. The other ligands, the other receptor might have much more complicated ligands to study. Having the TMB, the MSI, mentioned here, so tumor mutational burden can tell us, yes, this guy might respond to immune checkpoint blocking, but to what immune checkpoint blocking, to what combination, TMB will not tell us. This artificial intelligence probably will tell everything. I'm joking. But yeah, well, it's time to see. So we would like to envision that array of functional reporters for the various immune checkpoint pipelines will be important part of a toolkit that the physician will have to make the correct decision for the patient what treatment to get, that it will not be just personalized, but also precision cancer medicine. So thank you for the collaborators for the studies was published, mostly Karen Rubinov from Soroka, and Marshall Porevsky and Alexander Olivov from Barzilai, and Professor Salem from Rambam Medical Center. Thank you for my students. All is not in the picture, but most of the work was done by here, the MD PhD student, very talented MD PhD student, a blessing for the physicians, Bar Kaufman, and thank you to the other students of the lab, and thank you, Ronit, for inviting me. Absolutely, if someone, well, but then he should have also a mechanism to decide whether it will work or not, but yes, I guess that most of immunizations to get so many MABs to PD-L1 was done with purified bacterial produced, no glycosylation there. If someone would use it from eukaryotic cells, depends what eukaryotic cells you are producing, he might get much better anti-PD-L1. And probably it is done now, I guess, because in the market, in the clinical trials group, there are at least 20 clinical trials with 20 different antibodies to anti-PD-L1. So hopefully it's done. Great. So I'm repeating the question of Ravid. Do I incorporate the PD-1 levels into the score? Not yet in the formula, but we have the intensity, which, as I showed in one of the first slides, is usually reflect not the PD-1 levels, but the PD-L1. So through the intensity, and the reporter is 1, PD-1 is 1, so we don't need yet we need to improve the formula, I guess, but not with the PD-1 levels yet. Yes, that's a great question. As I said, the question is, can you evaluate 2 checkpoints? Not yet sophisticated enough, we are, I'm talking like Yehuda, to evaluate in the same reporter, but definitely we showed that we can evaluate combination using 2 reporters, one for, let's say, CTLA-4 and one for PD-1. So we believe that we can answer the question of combination, but needs to be tested yet.
You mentioned other like TIGIT, LAG-3, all
Yes.
those that are in clinical investigations or even approved, but still, even LAG-3 that was approved, we see that the main inhibitor is anti-PD-1 that work typically. And
It depends whether you are asking BMS or another company.
I'm asking the clinicians.
The clinicians might not like so much the Obdualac, but at least he is less toxic than the combination of EP and
My
Nivo.
question was, can your study maybe explain why anti-PD-1, this axis is more efficient for inhibition than any other?
Well, if we will get examples, clinical examples from patients that were treated with Obdualac, which is the combination of anti-PD-1 and anti-LAG-3 versus EP-Nivo, and put the 3 reporters that we already have, we might get the explanation, but we are not lucky enough, or there is not time enough that we have some samples of patients that were already treated, and the results are already, the response results of the patients is there, but this is one of our plans, maybe with BMS.
Thank you very much.
Thank you.
And thank you to all the speakers from the first session. We have another treat in the second one.